Biology Module Lecture 2, Reproduction. The first thing we're going to go into when we talk about reproduction is the cell cycle, which is comprised of 4 divisions. The first one we'll go into is G1, called G because it is growth 1. This is often called the pre-synthetic gap. During the G1 stage, cells create organelles for energy and protein production, and they also increase in size, hence the growth. In addition, passage into the S or synthesis stage is governed by a restriction point, or the checkpoint that you see. Certain criteria, such as containing the proper complement of DNA, must be met for the cell to pass this restriction checkpoint and go into the next stage. The next stage is the S phase, or the synthesis of DNA. During this stage, the cell replicates its genetic material so that each daughter cell will have identical copies. After replication, each chromosome consists of 2 identical chromatids, which are bound together at the centromere. After the S phase, the cell will enter the G2 stage, or growth 2. This is called the post-synthetic gap because it happens after the S stage. During the G2 stage, the cell passes through another quality control checkpoint, which is listed at your right, the G2/S checkpoint. This checkpoint ensures that the DNA has already been duplicated and the cell has enough organelles and cytoplasm to divide between the two. This checkpoint also ensures that DNA replicated properly and that there isn't an error that's being passed on to the daughter cell. The last stage is the mitosis stage. This is mitosis proper, along with cytokinesis. Mitosis is divided into the 4 phases: prophase, metaphase, anaphase, and telophase, which we will go into a little bit later in the lecture. As you can see by the divisions listed, interphase is a substantially longer phase than mitosis proper, and interphase, as we listed earlier, is divided into G1, S, and G2, with mitosis only comprising a very small portion of the cell's life cycle. Now that we've gone into an overview of the life cycle of the cell, let's go into mitosis proper. Mitosis is the most common form of cell replication that you will see. It's comprised of prophase, metaphase, anaphase, and telophase. A common acronym you can use to remember this is PMAT. Prophase is when the chromosomes condense and the nuclear membrane dissolves. The centrioles migrate to opposite sides of the cell. Prophase is listed right here, as you can see. Metaphase is when they line up along the metaphase plate, and the centrioles attach themselves via spindles. Anaphase is when these chromatids are separated and pulled along, while telophase is when the nuclear membrane reforms around the newly split cell. And finally, cytokinesis is when the daughter cells pinch off and split. Often, cytokinesis is listed as its own step. When it comes to sexual reproduction, you have to undergo meiosis. Note that mitosis creates 2 identical daughter cells, whereas meiosis generates 4 unique haploids, which are called gametes, and it requires 2 of these to produce a functional zygote. Often, this is called double mitosis because there's 2 phases of prophase, metaphase, anaphase, and telophase, listed to your right. A unique feature of meiosis is the potential to cross over, which is when the overlap of chromatids during prophase 1 will cause exchanges of genetic material. During meiosis, you will generate haploids, and haploids, as the name suggests, half-loids, instead of diploids, which are created during mitosis. An advantage of sexual reproduction over asexual reproduction is that sexual reproduction has a higher likelihood of creating genetic diversity. So we've covered mitosis, meiosis, and the cell cycle. Let's go into the reproductive system. In part, especially for the MCAT, the human reproductive system. Starting first with the male reproductive system. Sperm is the sex cell that's generated by males in the testicles. It's comprised of a head and a motile tail. Assorted glands and chemicals create semen before being delivered into the female via sexual intercourse. Spermatogenesis is the process of creation of sperm. Sperm oogenesis, creation. This results in 4 viable spermatozoa from spermatids. The female reproductive system, on the other hand, produces an ova, which is produced in the ovaries. This is sent down the fallopian tubules, where it will be fertilized and implanted into the uterus. Oogenesis is the production of ova, but it only produces 1 functional haploid. The rest are non-functional polar bodies. The diagram for this procession is listed to your right. The gonadotropin releasing hormone, or GnRH as it's commonly abbreviated, so GnRH, is released from the hypothalamus and triggers the release of follicle-stimulating hormone and luteinizing hormone, which have various effects depending on the gender. In males, FSH triggers spermatogenesis and LH will trigger testosterone production. In females, FSH stimulates ovarian follicle development and LH causes the ovulation. And take note that in females, both of these hormones stimulate estrogen and progesterone production. The last thing we'll get into in this lecture is the menstrual cycle. This occurs only in females. It's comprised of 4 steps in particular, the first being the follicular phase. This is when GnRH releases and stimulates FSH and LH secretion. Follicles then develop, and estrogen is released at this phase. During ovulation, a sudden surge in LH triggers the release of the egg from the ovary, and the luteal phase is when the follicle ruptures and forms the corpus luteum. This secretes progesterone and maintains the uterine lining. Lastly, menstruation. This occurs when no fertilization occurs. This is when the endometrial lining is shed. This cycle repeats about once a month, approximately 28 days, and it occurs if there is no fertilization. When fertilization does occur, you will have a different procession. This is something we'll go into in the next lecture, so don't worry about it for now. Alright, so we've covered reproduction proper. In the next lecture, we're going to go into embryogenesis and development, or successful fertilization. So, we'll see you then.